workshop is basically in this workshop we are focusing on a technology which we call as a light sheet technology, which is in a year to come. So it is a technology of the future. So I'm just going to give you some of the overviews of what's happening, some historical perspective, and what's happening right now. So let me share my screen, and we can go from there. I hope you're able to see my screen. Can one of you confirm?
Yes, sir, it is visible.
Okay. So fine. So let's get started. So as you see the title, it talks about light sheet technology. I call it technology because it has a lot of implications in many branches, and not just what we have seen till now. So what we have seen is mostly the applications are in biology. So I call this as the next gen technology, because you'll see it becomes obvious that technologies that are presently we have are basically point based. So what we are talking about now is the light sheet based. So these are the differences between these two basic technologies that are existing and that is yet to come. Okay, so let's move forward and have a look. Hold on. Right. So I will start with what we call as the light sheet tree. And this contains, most of the fields that I envision as the fields where, things can work out. So some of the fields, for example, optics and biotechnology, developmental biology, bit of medical science are basically the ones where a lot of progress has been made in the last decade or so. Out of them, developmental biology, I think, is the one that has benefited the more. So here, if you see the blue and blue lines, basically here represent that the field is kind of matured as far as light sheet technology is concerned. On the other hand, the green lines basically show that the fields are on the go, and a lot of things are yet to be done. And if you look at the red ones, where nothing is done, almost nothing is done, and a lot needs to be done. So as I say, that light sheets are for the future. There is something that is not yet been explored, and a lot of opportunities to lie for all of us. So if you see, manufacturing is something where machining and building are mostly done by point-based illumination kind of technique, which is not explored yet. Optics is quite explored, and nanotechnology is something that is just started to penetrate. Developmental biology is quite matured as far as light sheet technology is concerned. So it is extensively used. Marine biology is something that we look forward. A lot of industrialization of light sheet technology has not been done. The one that we're using now is what we call as a point-based technologies, where we'll talk about this more. So device and instruments is something that is yet to come. Biomedical devices is a big area where a lot of work has to be done using this technology. And if you look at biotechnology, things have started to roll out as far as cell biology is concerned. And drug discovery is yet to come. Actually, this is something that is done in bits and pieces. Now, look at medical science. A lot exists there. And there is some process has started where biomedical devices, cytometry, imaging, endoscopy, these are something that has been proved. But clinical research is something that is yet to be done, and it is so big, so huge a branch it is. So anyway, now you have an idea about, we have an idea about what all things are done, what all things are need to be done, where the opportunity is basically live. Okay, so let's see some of the milestones that we have here. I haven't taken care of all the milestones that here. It's a little hard, so I've taken this from one of the papers. So the light sheet has light sheet microscopy, or you can say light sheet is started by Seidentov and Zimondi in 1903, and they called it as ultra microscopy. Then the technique was forgotten for almost 90 years. And in 1993, off-force microscopy is proposed by Voey and Spellman, who reinvented, I would say, or brought back the light sheet. Again, it was forgotten for 9 years. Then Stelzer's group started the light sheet in a big way, and they called it, selective plane illumination microscopy, which will be also one of our speakers is going to talk about. So it started here, and then it has grown quite fast. And then you have 2008, another, this digital light scanning microscopy has started, and then you have vessel beam light sheet microscopy 2010. Then structured illumination in 2010, super resolution, which also you're going to look at, 2-photon light sheet. A lot of variants have come over time, multi-view light sheet, confocal light sheet. But then something happened in 2013 where people have looked away from applications in biology. So now they're looking applications in engineering and physics. So here you see beam shaping. So Lashkin, for the first time, tried to demonstrate beam shaping in 2013. And then in the same year, our group also started a light sheet cytometry, which is a big part in medical imaging or medical science. Again, Wu's group or Chan group has started light sheet cytometry, but in millifluidics in 2013. And then you have, then this lattice light sheet by Bedzik's group, and then you have Keller group, which talks about ISOView light sheet. So all these things have kind of, bloomed up. And beginning 2013, we started to see some applications in physical sciences or engineering sciences, I would say. Okay, then if you go a little forward, you will see that in 2015, the first application nanolithography started using light sheets. So we call it I-class nanolithography, which actually started in 2015 and it's still growing, but most of the application, most of the technology is being shown till 2020. Then 2015, again, optical magic carpets. This falls in optical physics by Golub. So we're going to talk about this a little bit more. Then 2017, organism level screening, of light sheet, in the flow is something that also was shown. 2021, Pi group has shown application in plasma physics. So I'm going to talk about this briefly as well. But in 2021, again, light sheet optical tweezer or traps, optical traps, is also shown. So basically, the idea is that 2013 onwards, there are applications which are actually emerging in physical sciences. Before that, it was predominantly biomedical or biological applications. So we can see that now the light sheet is penetrating both physical sciences and engineering sciences. Okay. Right. So let me at least get the basics a bit clear about how we look at it. So existing technologies, as I talked about, the point-based technology, you have a focusing element here. And you can see the light gets focused and, you look everything at the focus. So this is the focal plane. And what you observe here, while your focal plane, is illuminated, you have also illuminated off focal planes. So, which is a concern when you do sensitive imaging, using fluorophores that get photo bleached. And this is kind of unnecessary that happens, but there is no selectivity there. Yeah, this is one of the limitations of point-based illumination. On the other hand, if you see sheet-based illumination, you illuminate from side. So this is the whole specimen. You illuminate from side and you select a specific plane. And what you do in the process is that you have a selectivity. And you don't have unnecessary illumination of focal planes. So this is the advantage that you see. So in as you can see here, you have the specimen, you can illuminate from side, not from the top. So, and 3D, this is how it looks like. So the basic idea is that we are moving away from point-based illumination to sheet-based illumination. And, a lot of advantages we see. And, of course, there are some limitations as well, but we're going to explore light sheet more. So this is the basic idea about it. Okay, let's have a relook at the existing point illumination systems. Okay, So if you see existing technologies, if you go for, if you look at extreme engineering where people use welding and laser machining, they are all done by point illumination. The question is, can it be done by light sheet based illumination? Again, engraving, machining on metals, laminates, acrylic. These are all extreme engineering applications where, again, point illumination is being done. Laser printers, you have point illumination. You have 3D printers which print organs, small body parts, houses, as well as, you can see all these small kind of things. So every field it has penetrated, but most of them are basically point illumination based. So we can say that point illumination based technique, point illumination is at the heart of today's industry and research. Now, question that we're trying to ask, can light sheet help, or can light sheet progress this or further this? That's the point. Okay, it's always good to look back and you can see that the first light sheet, as I said, it was in 1903. And the first Nobel Prize was awarded in colloidal chemistry in 1925 to Zimondi and Sedentov. And this is the basic, structure or basic microscope that they have made. So you illuminate from side and you look at from the top. And what they looked at as the first application, looked at the gold colloidal particles. And they used their illumination as sunlight and split to, to realize the sheet. And if you look at today's present-day developments, you can see most of the present-day microscopes are based on this. So basically, you can say the most of the present-day developments in light sheet are basically improvisations or automation of this monumental work. So, and you can get the original paper here with detail. There are more details. It's a wonderful paper. You can actually go through this. But the idea is that the idea was already proposed in 1903. And that is still going on. So the microscopy, there is nothing vastly that has changed as far as the basic fundamental principle of microscopy is concerned, design is concerned. Okay, so let us look at light sheet for biological sciences or biosciences, biomedical engineering, bioengineering, or biotechnology, whatever. But this is something, light sheet has something shown is worth a lot in biosciences. And we are going to, look at some of them, not all of them, some of them. So as far as plant science is concerned, if you look here, you see, during the early development of the plants, you can see this is like a small plant. People have looked at the root development, and you can see one of the examples here. They've seen the cross sections. So fluorophores, for example, dyes or fluorophores are used to label different organelles, which people want to look at. And then they probe this with light sheet, and you can see the structuring of the root as it is growing, and you can see all the different, structures by which is labeled by different fluorophores. You can look at the leaves also, where you can look at plastids, which are GFP labeled. So it is the green fluorescent protein labeled, and you can look at the plastids. So this is something that is that has actually been very exciting in plant sciences. In another example that I have taken, you can look at the plasma membrane marker is a plasma membrane of the root section of a you can see a root section of a plant. Where you can look at, it is labeled by GFP, green fluorescent protein, and you can look at the nuclear markers, which are labeled by red fluorescent protein. The idea is, the idea here is basically to study plants using light sheet microscopy, which seems to be the most sensible thing, because you want to look at large area. That's something that is possible with light sheet kind of technology. Okay. So here's something that we have done in the lab, and this is done by one of PhD students, Jigme Basumatari. And here we are looking at Arabidopsis plant, and the stem section we are looking at. And the microtubules in that stem section is labeled by GFP. And you can see the life as it moves on, as it goes on. I mean, this is something that really is very exciting. And you can see the working of this, microtubules. and they're carrying the cargo, moving around, all kinds of activities that's happening inside the cell. Inside this stem section of the Arabidopsis plant, and this is something that is quite a exciting thing to look at. Right now. In developmental biology, light sheet has been extensively used. For example, one of the examples I can give you is Drosophila. So you see there are various stages of Drosophila, stage 5 to stage 17, that that is starting from 1 hour 40 minutes to about 15 hours 50 minutes, and you can see the developmental activity that has gone through the process. And this is something that Houston could show in 2004. Using light sheet, and you can see as it goes from stage 5 to stage 11. Stage 11 is somewhere you can see the structuring is happening, okay, and then stage 13 you can see the emergence of eyes. So that eye organs are starting to develop, and thereafter you can see various different organs basically come up. So one of the fundamental questions in developmental biology is which organ develops first and why. So there are certain things, certain organs develop before other organs, and there is a kind of synchronized development that happens, and this is something that is visible through this this work that is done by Houston. Okay, something that we have also tried to do in our lab also is so you can look at the muscle development in zebrafish. And you can see at 0 hours, you don't see much of, the twitch that is labeling the muscle. And during 4 hours, maybe little development, little changes, but in 9 hours, you can see the muscles have kind of developed well. So you can, as you look at through the organism at different stages, live organisms, you can actually see through light sheet the development that has taken place, and you can quantify them. So if you go through this book, you will see that they're quantified also using some metrics. So the idea is that you should be able to see the development over the entire organism rather than on a very small, very small region, or for example, in a cell. So that is like quite limited. So here the light sheet gives you a broad field of view. That is very, very, very important. You can see large specimens. Okay, then there is another interesting example of that of C. elegans, where people have drawn the neuronal map throughout C. elegans body. You can see head section, mid body, tail, and they have used more than 4 or 5 fluorophores to label different organelles, and they could show the dynamics. And this is all the study that we have done on live specimen without any kind of constraint, which is the best possible scenario as far as the biology is concerned. So most of the time, what people do is they, stick the C. elegans with a glue or something. They constrain them, but this is something where they have actually overcome that, and they could see using light sheet and that all the sections and as they develop in their developmental pathway. Okay, so if you go, so there is another application where light sheets, where C. elegans are made to flow through microfluidic chip, and in the process they are being imaged, and you can see different sections of the C. elegans. 1, 2, 3 sections are actually there. Many sections you can look at. So this is something that is you are also going to do in today's workshop, and so this system is already there in the lab. You can actually work on this, and you are going to make this system by yourself. Okay, now coming back to tissue and organ biology. So light sheet has a bigger role to play, and it is playing a big role because it can go from cellular level to almost organ level. So you can see here lungs lobes of a mice, and this is uncleared section, and then some chemical processing has been done so that it is optically cleared and light can penetrate. And when light sheets penetrate, it looks a lot of things. It looks like this. So you can look at basically the structuring that has happened inside these organs. So you can see the veins, all different kinds of blood vessels and all. So you can look at them, and you can go multicolored. So if you want to look multiple things, that also you can do. So what altogether this is that light sheet can give you multicolor, It has all the properties of normal microscopy, and it can also give you a large field of view. You can investigate large organs. Another organ that people have shown, and that is shown by Keller group, is neuronal activity or zebrafish brain. So you can look at the zebrafish brain, and you can also look at the neuronal activity, which I'm going to show you in the next slide. But you can see the structure of the whole zebrafish brain, and as they grow, this is something live that's possible. Again, here you have the songbird brain imaging that has been performed by Rocca, and you can see the structures inside. You can go multicolor. So the idea is that you can go from cellular level to organ level, which is a 10 to the power 6 orders change, okay, from micron to centimeters. Okay, so as far as neurobiology is concerned, people have looked at neurons and neuron firing and all related activities, as well as they have looked at calcium movement inside, in the neuron. And they have labeled this calcium with a fluorophore, and they looked at it, and you can see the fluorescence. And this region, many regions they have selected where you can actually look at them. And the good thing about light sheet is that you can look at very large area. You can look at multiple neurons or multiple sections of a neuron. So a large area you can look at, and you can quantify them. You can find out when it's firing, how it is, how the calcium is, is moving from one part of neuron to another part. So all these things you can look at it. Then you can look at different sections of the brain with a diffraction limited resolution, and you can see dorsal view and lateral view of a zebrafish brain. And you can do functional imaging. That's the important thing. You can do functional imaging on live specimens, which is something that is not possible with existing technologies that much. It's hard to do, but light sheet enables you readily. Okay, so these are all the applications that are there in biology or biomedical, bioengineering. Is kind of related to biology. The question that you're asking, how much it is useful in other fields. So we'll see that the light sheets have arrived in physical sciences as well. Okay, so first is the light sheet in biomedical physics or engineering. So you can see you have a microfluidic chip through which you can flow the cells, and you have a light sheet that is sectioning the microfluidic channels, and you can look the fluorescence at orthogonal to from the illumination. And you can see, you can, as the cell passes through these multiple channels, you can look at different sections of the cell. So you have, this is one cell, this is all one cell, and you can, we got, something like 5 sections. You can get 5 sections out of it. And if you put together, you can actually, look at the entire cell. Now the cell is flowing, so it is actually spherical in shape. And you can see all these dotted structures, red color structures. These are all mitochondria in the cell, distribution of mitochondria in the cell. So the advantage of iLife system is basically this is optically very simple, which means it can be manufactured without much of an error. It has multi-purpose diagnostic possibility. It is highly automatable, economically sustainable, commercially viable, and it can be a complete healthcare solution. So as you can, you get something like diffraction, near diffraction limited resolution. And that essentially means that existing microscopes which are used in hospitals or clinics can be replaced by, cytometry system, iLife cytometry system, which is as good as far as resolution is concerned, as far as quality is concerned, image quality is concerned. What all kinds of studies we can do? We can do whole population study, that includes cancer screening, disease screening, hematology, pathology. So, and you can do fundamental studies on multicellular organisms as well. So this kind of system is basically, you can say, all in one kind of system, which if tuned properly, and if it is, little bit more practicality, if it is included in it, then incorporated, then I think this can become the next generation diagnostic system, and a single diagnostic system which can do a lot of things. Okay, now we move on to material science. So light sheet has also found its application in materials, specifically related to nanolithography or lithography, you can say. So what has been shown is that two opposing, light sheets, PSF, that basically, have the same focal area. And as you know, that interference is bound to happen, and you can get what you get is standing wave-like pattern. This standing wave-like pattern, if you put a specimen, your photoresist or photosensitive material, then you can actually, develop it, and you can get this kind of nano channels, which are of 350 nanometer size. And, nanofluidics is something that gets boosted by this kind of, this technology. So this technology is very simple and robust. I would say this is very initial stages. We are actually at the initial stages of this. And, of course, we're looking forward to have more and more applications. So this technique, this nanolithography technique is simple and robust. It is miniaturizable, and this is highly on-site fabricable. So that means in space, if you need to fabricate something, this kind of technologies are very, very useful. There is a paper also by Stelzer Group on application of light sheet in space. So that is something that is worth looking at as far as light sheets are concerned. So then you have the good thing about this kind of technique is that layer selectivity. That means given a 3D volume, if you have a 3D volume, you can actually select a specific layer which needs to be, which you want to imprint something. So you can selectively, you can select your layers to be imprinted. And that is something that is possible with the light sheet. So layer selectivity is a very, is a strong point of light sheet as far as, nanolithography is concerned. So as I said, space applications are there. So then what are components that has been realized? As of now, we have realized nano channels, nano waveguides, nano gratings. So this can be used as this kind of, nano kind of gratings or waveguides or channels. Nano chips is something that is on the horizon. We haven't done much of it. So nano chips is something that we're looking forward to, and this is possible because of the selectivity. Okay. Now, there is something else has happened also in physical sciences, and there is in optical traps or optical tweezers. So using light sheet, we actually, in our lab, have successfully shown that it can be used as an optical tweezer. And we have trapped dielectric beads, we have trapped cells. And this is something that is very, very simple. As you can see the geometry, you have a cylindrical lens, and you are followed by objective lens to make a diffraction limited light sheet. And this is where you can trap. This is the PSF, system PSF, where you can trap all the particles. As you can see it here, I'll show you later on a video as well, which gives a better idea. So let us look at the advantages. Optical levitation is possible, and line traps, as you can see. But another thing that we haven't done yet, and we are, as I say, the work is in progress, we haven't done yet, is what we call as the plane trap. So planar trap. So you can actually trap particles in an entire plane, entire layer. That is something that is yet to be shown. Then live cell patterning is something that can be done. I will show this later on. Then you have 2D studies that you can be performed, and you can do feeble force measurement on a plane. That is something possible using this kind of specialized light sheet. So specialized light sheet with optical tweezers. Now, as I say again and again, these are all the fields that are just opening up, and nothing much has been done. A lot needs to be done, and this is something that's the opportunity build up at the time. Applications that we have seen envisioned is basically cell-cell adhesion studies we can do. Life cell patterning and growth is something that we are able to do, but more we need to figure out. So a lot of application is still unexplored. So as I say, the light sheets in traps are unexplored. This is something that is yet to be explored, and we are just at the beginning of this. The same is true with in material science, nanolithography. The same is true with imaging cytometry as well, or medical science, as I say. So the field of medical science, material science, optical physics and optical tweezers, as I say, are basically the fields to look at. These have just started to bloom up. Okay, if we go deep in optical engineering, then people have also created what we call as magic carpets. These magic carpets have you can see the illumination is basically quite strong, along the Z direction, and this is just at the focus. And if you move away from the focus, you are supposed to see a diffracted beam, right? So, but these magic carpets have the property, they are diffraction free. So if you go even 100 microns far from the focus, you still see that it is not diffracted. This is very different from what happens with Gaussian beams. Okay, so this have applications in deep penetration, large area investigation, and since they are diffraction-free beams, they can penetrate at large distances. Applications are into photon microscopy and brain imaging. And all the the related publications I have given at the bottom, so that you can look at. If you have more interest, you can move forward and look to those publications. Okay, another application is optical beam shaping. Ideally, what we want in microscopy or in any kind of imaging is uniform illumination. We really don't want a non-uniform illumination, but that seldom happens because the beams that we use are Gaussian in nature. So we have Gaussian beam here, and this is what we use for all kinds of imaging in microscopy. But idealistic beam should be like this, which is uniformly illuminated. And this is something that is possible by what we call as the pi shaper. So this is something that is commercially available and that uses cylindrical lens to prepare a sheet kind of technique. So the good thing about this is that uniform intensity investigation can be done, which is the idealistic case, and large area interrogation is also possible if it is associated with cylindrical lens to make a larger light sheet. What are the applications that people have shown? The applications are in particle image glossimetry, laser-induced fluorescence, hardening, annealing, all kind of engineering applications people have kind of done with light sheets. So this is just to show that light sheets has already penetrated physical sciences as well as engineering sciences. I can say mechanical sciences as well. So in a look there. Okay, one of the interesting applications that I've seen of late in physics is basically the plasma physics, is the detection of short-lived aqueous species, which is OH, in one of the experiments, and that is possible using the light sheet. So they have exposed the light sheet at the interface of two liquids, and they have looked at the interface, and they could find out that short-lived species, which are otherwise hard to look at, hard to recognize, can be actually detected. And you can see OH species, H2O2 species, NO2 minus species, NO3 minus. So all these species, aqueous species, are basically investigated using light sheet spectroscopy, I would say. Okay. Now, all these things that we have known now, that now things have already progressed and moving forward in various directions, specifically in physical sciences. Now, I'm going to talk about some of the basic things about light sheet here. So we'll look at the variants and current status, because you need many different kinds for which is very, very suitable for specific application. So we have many enriching configurations that are possible. This is called open top SPIM, where you have the excitation, and you have direction at orthogonally, which is in 90 degrees. So your specimen is somewhere there, and you can illuminate and you can look at. Then you have tilted light sheet system, where you have excitation from one of the sides and used a prism to really focus that. The light sheet here, and you can look at the emission on ortho perpendicular to that. This kind of same side, not really, but you can say it is angular kind of. There is an angle between illumination and emission. Dual illumination, you can illuminate image, again illuminate image. Okay, so this is something that is possible. Remember, this is an inverted system, this is the upright system. So all kinds of microscopes support this kind of technique, light sheet technique. Single objective SPIM is something that is really nice because you don't have very cumbersome design, and you can actually illuminate and image both using a single objective. So illuminate from side and you get a light sheet here, and that can, the fluorescence, whatever light comes up can be collected by the same objective and you can look at them. Diagonal spin is something really good, and this is something that the participants are going to do by their hands in this workshop. So you can see your, excitation is and illumination. They are illumination, excitation, both. Sorry, illumination and emission, both are orthogonal to each other, but a specimen is at an angle. That's why we call it diagonal spin. Then you have inverted spin. Then you have oblique plane illumination, where you excite from one angle and detect from another. Reflection light sheet. So what I mean to say, there are many configurations that can be used, and that is maybe suitable for specific applications. And so diversification has already happened of light sheet, and it is just that one has to use it for specific application as required. What are types of light sheet we have? So we have the classical light sheet, where you can actually use a cylindrical lens and you can generate a sheet of light, and you have orthogonal detection. Then you have what we call as scanning light sheet, which means you actually generate a pencil beam, and then you can scan it so fast that if you scan it very fast, so it appears as if it is a plane. And this is what we is called digitally scanned light sheet system. And this is a different kind of generating a light sheet. Then another kind is what we call as Bessel light sheet. Okay, so normal light sheet, if you look at Gaussian light sheets, which is also the classical light sheet, it has a finite extent. Bessel light sheet, the extent can be enlarged, so extent can go large. So you can penetrate deep and you can penetrate large, big. You can penetrate deep and big, and this requires as Axicon as one of the basic components. And you can use a galvanometer to really scan this and make a large, field of view. Not field of view, large section you can scan. Okay. There is something else that I miss here, is what we call a curved light sheet, which I haven't somehow not included. The curved light sheet is something that is kind of proposed and that is kind of trending as well. Okay, now we'll talk about one of the applications of light sheet, which is taking the note of many, which is getting noticed by a lot of researchers. I would imagine as of now, my information is more than 20 groups, 10 to 20 groups are something that have already actively working on this. And that is eye life cytometry system. Okay, so this is the light sheet cytometry or eye life cytometry system. So the beginning, as I said, we started this in 2013 on microfluidics, and Wu's group has started this in minifluidics using a capillary. And we could show that different configurations are possible. You can see this is orthogonal configuration. This is not orthogonal. So this is pi by 3 configuration. As you can see, this is pi by 2 configuration. Then you have organism level imaging, that is something that is shown. And then, the people have studied vesicle medium and all. So I have seen some papers also coming up on imaging cytometry in Nature and Science journals, which are like quite big journals, I would say, and it's getting the prominence that is required. Then you can look at, as I showed you, you can look at cells as they flow through, but you can do it through many channels as well. So this is something that we're going to do in the lab. Of late, what has happened starting 2017, PDMS micro devices have come up where you can have the whole light sheet on chip. That is something possible. And your microfluidics and light sheet both are on chip. And it's like you have a micro device available that can be used for various applications. So the commercialization has already started as far as eye life imaging cytometry is concerned, or imaging cytometry is concerned. Then there are much diversification has happened and micro devices. You can look at some of the micro devices that I have taken from some different papers, where they have integrated microfluidics with light on site. So there is no separate illumination. It's all embedded in a single chip. This is what people have achieved. And you can see different papers that have been published where they have talked about this and they've realized this. So, okay, light sheet has another application as far as imaging is concerned, and that is it can go very fast. It can go very fast. So what we have observed, we have observed 10x times faster than traditional imaging. and that is based on rotation. So you have a sheet that can, that can give you, you have a light sheet that you can scan in a rotation, in a circular manner. You can rotate the specimen and you can look at different planes, and you can see some of the planes that we have looked at of, zebrafish, where we have looked at the microfibers as well as macrophages, and you can see the images of microfibers and macrophages. This essentially means that you can have very good resolution, and at the same time you have a better temporal resolution, which means you can go much faster than what is possible. That is what is being shown in this application. And here, multicolor is something that is explored. Multicolor light sheet for volume imaging is something that is explored. And you can see this multicolor, where blue, and you have a single PSF, which we call as the multicolor PSF, and that can be scanned throughout the system. Time-lapse imaging is something that that we have also shown. This is also shown at 0 hours. You can see the structuring, and at 9 hours you can see the structure. I'll show you a video also of this. And then there are some fundamental studies that can be done. So we have looked at the lipid-dependent metabolism and protein-dependent metabolism and the crossover that happens. So all these things are kind of reported, and you can see at different periods. At the same time, all these things you can see it at a very rapid, in a rapid pace. Basically, you can do the volume imaging at very fast. Okay, here is the time-lapse video of the muscle development. Some of the planes that I'm trying to show you, I can look at how the muscle development has happened. So as it develops, as it goes through, I can see the structuring that is happening within this one, right? So the idea here to show that you can actually go live, you can go fast, you can look at the entire volume. That is something that is very unique to light sheet microscopy, as when you compare to other kinds of microscopy like confocal or even super resolution microscopy. Although there is a super resolution analog that another speaker is going to talk about. So what all the future directions are, and I have figured out some of them, and there are many more possible. So here you can look at what we have done. We in clinical diagnosis, clinical biology, we have looked at. So if you see the cancer biology here, you have the HeLa cells, and we have looked at the mitochondria within the HeLa cells. You can see they are all randomly distributed within the HeLa cells. That is how it is supposed to be, kind of, not really random, but there is a kind of particular pattern is there. But when you look at all over, it's there, the HeLa cells. Now, when we treat it with a drug, and what we have seen that the HeLa cells, the mitochondria is essentially deposited, or it is actually moved out. It has disintegrated. Mitochondrial disintegration has happened, and this has moved out to the periphery of the cell. And this represents a cell apoptosis, which means cell death. And we have also done mortality studies, which means we have taken the HeLa cells and we have treated with the drug. And what we have looked at different concentration, what we can see that at higher concentration specifically, more, so here orange color represents dead cells. So more HeLa cells are dead. Okay, cancerous cells are dead as compared to, if you look at a small fraction is still there. The same drugs treatment studies we have done on HEK cell lines, which are healthy human cells, HEK cell lines we have done. And what we have looked at, what we have found that at higher concentration also, the cells, these cells are actually alive. So which means that we are selectively targeting only HeLa cells that can be, we can actually selectively target them as far as the cancer biology is concerned with this drug. So this is quite an exciting thing, and this is all studies is done by based on light sheet. That's what we call as iLife system. Okay, and as I said that I will also show some applications in optical tweezer, and you can see this is the basic diagram as we know, and you have used this light sheet to really trap cells, HeLa cells. So you have trapped HeLa cells, and you can see, look at 1, 2, 3, 4 HeLa cells you have trapped. Here we have trapped 3 HeLa cells at an angle, at different angle, and you can see vertical. So we can rotate the light sheet Just by rotating cylindrical lens, you can rotate the light sheet and you can trap different. Using this, we have written IISc. You can see here, IISc, and using cells. You can write other, structures as well. For example, here we have written kind of C and T, and you can see that C and T are written. Not only that, we have actually looked at their development, which means we have looked at their growth. So you can do growth studies. So you can put the cells in a specific pattern, and you can actually study or see how they're growing over time. And you can see till this it is T, but after some time you can see the growth is kind of irregular. It has become. Similar things we have done as far as our geometries are concerned, geometrical objects are concerned, in a line, in a triangle, in a Z kind of shape, and we have seen how they have grown. This work is in our lab is done by Neptune Baro, Jigme, and Joshi, perhaps Prakash Joshi. So kind of it's a group effort that I've done, but yeah. So optical tweezers are something that has a lot of potential as far as different applications are possible. And this is not only limited to optical physics. We have shown some of them. A lot of people have shown a lot of some of them, some of the fields, but it is not totally completely done. Okay, let's have a look at how the light sheet is able to trap. So here you have the dielectric beads. Some of them are trapped. I will look at how it goes on. And you can see how it is trapped. The light sheet is trapping one by one all the beads. These are all 2 micron dielectric beads. And as I say, you can make any kind of structure possible with this. Okay, what are the future potential? As I see, future potential are multiple light sheets. So we know one light sheet. Can we have ways to generate multiple light sheets so that you can parallelize system, you can have faster system? And a couple of ways we figured out is basically spatial filtering technique and grating-based technique. So in spatial filtering technique, you have many light sheets, but you can see the structure of light sheets. They are actually gradually going down, whereas much more uniform structure of light sheet you can figure out using grating-based technique. This is also, power-wise, energy-wise, this is also efficient process. Then another, future potential is in Bessel beam, Bessel light sheets. So Bessel light sheets are really, really long, and they're diffractionless beam, which means they don't diffract. And another good property, another wonderful property of Bessel beam is that even if you disturb, or even if there is a microparticle or something scattered part, a scattering object here, it can rebuild itself. So that means it is very, very good for depth imaging. Extended light sheet is something that can be used for large field of view imaging, is also one of the futuristic things. So these are futuristic kind of light sheet which we can look at, and that's going to come up in times to come. Okay, now let's have a recap of how the fields are moving. So what is the present status basically? So this is the approximate idea. So I have looked at, and you see health sciences. So biomedical and clinical biology is something that is kind of started. Whereas if you look at biological sciences, this has benefited the most. As far as developmental biology is concerned, it is huge. And one of our speakers is also going to talk about how the development happens, developmental biology, light sheet in developmental biology. So cell biology is also benefited. So you can see biological sciences is basically booming with light sheets, structured in biology, biophysics. But engineering sciences is something that is lagging behind. So you can look at computational sciences, bioengineering, biotech, engineering sciences. The applications or the interest is basically low. If you look at physical sciences, optical physics is something that has grabbed it in a big way. Light sheet is being used in a big way. But other than that, if you look at nanophysics or material science, plasma physics, it is yet to come. They have just started. It's yet to come. So there's a lot of opportunities in different fields, as I say, and it is just the beginning of something revolutionary, I would say. Okay, I'm kind of close to end of my talk, and these are the lab members. We have Prashant, Prakash, Jigme, Arvind, Neptune, who help me do different stuff. They do the hard work, and I really talk about their work. So, they are the one who's behind them. And we have a postdoc, Zibi, who has started a few months back, and she's doing well. And I hope that we can take this forward. So a lot of this, my students and postdoc, they are actually helpful in what I show you here. Most of the results are basically done by them. And these are the guys who are going to, are going to show you how to build up the system, how to understand the biology behind. You can go to Zibi, she's a biologist full time. So you will know a lot of biology, you know a lot of, imaging, and you know how to make your own systems. So this is what is the goal of this workshop. And you are going to fulfill this goal. I know when we look at this, fine. Collaborators are always important because they are the one who keep us going. So we have Professor Alberto Diaz-Pro, Upendra, Abhishek, Peter and Samuel he has been quite a close friend. Francesca, and I would like to mention here, the first time I did light sheet, it was with Francesca. She's the one who actually told me how the light sheet works way back in 2011. So, I have all kind of people who have kind of helped me on my way. And then we have funding agencies with whom without them we can't move forward. And we have funding from MHRD, DST, SARV, DBT, INSA, BRLS, all throughout the last 10 years. We have got good support from these funding agencies, and we are really happy that they moved forward. They came forward to give us all the support that is financially required. Okay, I think I'm kind of done. Here are some of the review articles and books that you can look at. So these are some of the articles that I've seen, which is like really, really good. And, they give a comprehensive overview of what's happening in the field. Okay, you should look at this Nature article, which is just published in Nature Primer Journal. That talks a lot, and this is here one of the article. Look at Frontiers. This article is really, really good, where I got a lot of update on what's happening. Okay. Anyway, and you can have a copy of the book, which is very rare. Rarely, and there are not many books. I would say there are 2 to 3 books that are there in the market. And our book is one of the books which takes students from very basics to the advanced light sheet techniques. So this is something that is going to have handy. You'll have. Okay, so I've kind of done. And now, if there are any questions, people can ask, or just please go ahead and feel free to ask questions if you have any.
Do you have any questions? Anybody has any? You have one question.
Okay, you can ask.
Yeah. So if there are no more questions, then we can go ahead with the workshop.
Sir, one.
Yeah. Yeah, please. Go ahead. Go ahead. Go ahead.
Right, like NO3 minus and OH minus. Can you enlighten a little bit more about that part, about how to detect short-lived species and all?
Right, I will do that. Well, I have, so here you can see what they have done. They have 2 layers of liquids, water along with another liquid. And what they have looked at, they want to look at the interface. So they don't want to look in the deep. They want to look at the interface where these 2 liquids actually, come together. So they are not mixing. So these 2 liquids don't mix with each other. And what they have done, they have taken the light sheet and looked at this plane at the interface. And what what light sheet has done, basically, when you select a specific layer, so you have a good signal to noise ratio, which means you get good signal out of this, and which you can't get otherwise using other optical microscopy techniques. So they have used this light sheet, and they have looked at the Raman signal at the interface. From the Raman signatures, you know that Raman gives you vibrational information about the species. They could figure out that they can see that OH species is something that is short-lived, and they could actually detect them through their detector, which was not possible using other microscopic techniques. It is basically because light sheet can selectively illuminate a specific plane where this activity is happening, where these changes are happening at the surface. So these are surface physics is something that is being explored.
Thank you, sir.
So anybody has any more? I think no one has any question anymore. Then we can end the meeting.
Right. Yeah. Thank you. Thank you. Thank you for your attention.